Good morning, students. I am Dr Shrabani Mukherjee, lecturer in Botany, Marwadi College. Today, we'll learn about artificial vegetative reproduction in plant. It is the extension of lecture 3. It is lecture 4. In the previous lecture 3, we have already studied about the vegetative reproduction in plants. The various types of vegetative reproduction is man-made. They are developed by plant growers and horticulturists for commercial production of crops. They are called artificial methods. As they are man-made, there are many types of technologies. Hence, they are called artificial methods. Number one is the first method is cutting. A small piece of any plant organ used for vegetative reproduction is called cutting. Number two is layering. The stem branches are chosen there, bent towards soil, using auxins in the injured branch to form adventitious roots. Separate the rooted branch from mother plant to get a new plant. This is called layering. Number 3, grafting. This is the another method of artificial reproduction, that is grafting, the most common artificial method for propagation. The all fruit plants are made by this method. In this method, a small portion of stem of one plant called scion, SCION, which is inserted on the root system of another plant called stalk. So students, in this method, we have to take two plants, one for shoot system and another for root system. The shoot system plant is called scion and the root system plant is called stalk. The stalk develops as mother plant and scion develops as shoot system. Example is scion of high quality roses are grafted on wild rose root stalk. So when we see a very beautiful developed rose plant, the we can see that is the shoot portion which contains very beautiful flowers can be made by using this grafting method, by forming many scion, and the root portion, that is the very normal rose plant which is found in villages and contains very small flowers. So to get big flowers, beautiful flowers, we have to join two rose plants. One is scion and another is stalk. Number 4, micropropagation. Modern method of vegetative propagation takes place inside the modern laboratories called micropropagation. It is based on tissue and cell culture method. A mass of undifferentiated cells are produced by this method called callus. So, what is callus? Callus is a group of cells which is never differentiated in any organs or any tissues. They take differentiated into number of plantlets. Now, each cell of the callus give rise to a new plants. These plantlets are transformed to different pots or nursery beds. This technique is useful to obtain disease-free plants of orchids, chrysanthemum, gladiols, papaya, etc. Now the importance of vegetative reproduction. Number one is seedless plants can be easily grown. So students, you can see that banana fruit don't have any seeds inside the fruit. How can we propagate the banana plants if there is no seed inside the fruit? So banana plant can and can also under vegetative propagation to give new plants. Number two, the plants which cannot produce viable seed or are propagated by this method. That means the plants which have fruit and seeds, but the seeds are not viable. That means the seeds cannot germinate to give rise a new plant. Hence the propagation of that plant only by this vegetative method. Number 3, easy and less time taking method. Very easy because we don't have to wait for the time, for the season, for this method. Number 4, disease free plants are made by tissue culture method because the tissue culture method is always cell culture method. That's why a cell can be disease free. Hence, the full plant will be disease free by the tissue culture method. Thank you, students.